Wessinger from the University of South Carolina, whose lab uses the repeated evolution of floral traits among Penstemon wildflowers to study the genetic basis of adaptive traits. So please help me welcome Professor Carrie Wessinger. Also, the time after this and Pacific 318.
Awesome. Well, thanks so much, Aiden, for the invitation. And for all of you that took time to meet with me, it's been a really awesome set of meetings and a really fun visit. Okay, so please let me know if you can't hear me, and I'll be sure to project. All right. Oh no, there it is. Okay. All right, so many adaptations are complex traits where multiple components contribute to a shared organismal function. And we know that certain complex traits evolve frequently in some groups of organisms, but never evolve in other groups. And a familiar example of this would be the three-spined stickleback, where transitions from marine to freshwater have occurred many, many times and involve this complex but predictable set of changes to morphology and behavior. And so these repeated shifts are clearly the result of natural selection, but there are lots of small marine fish that have never invaded freshwater lakes. And so we're kind of left with some outstanding questions. So the first is, what enables repeated evolution in complex traits? Are there genetic features that predispose an organism to evolve a particular trait? So for example, maybe the adaptive shift can be generated through relatively few genetic changes because maybe individual mutations have large effects on the trait experiencing selection. Maybe they can be generated easily through new mutation because they have a large mutational target size. Or maybe they're already available, segregating in the population as standing variation. They might be recurrently introduced into a new population through migration events, and that could enable the repeated evolution through adaptive integration. And then, of course, how are these complex adaptations maintained in the face of gene flow? And this can be relevant when sister taxa have evolved divergent complex traits. So for example, what are the fitness effects of trait intermediacy? And so cases of repeated evolution kind of offer us the opportunity to study the origin and maintenance of complex traits using a comparative framework. And so we've been developing these Penstemon wildflowers as a new study system to understand how genetic and ecological features contribute to patterns of repeated evolution. So Penstemon is a large North American wildflower genus, perennial wildflowers of roughly 300 species. They've rapidly diversified. Diversity is concentrated here in the Western United States. And they show ecological diversification in lots of traits, including widespread patterns of repeated evolution. And a defining pattern is parallel evolution in floral pollination syndrome. And pollination syndromes are basically suites of floral traits that attract, reward, facilitate efficient pollination by a particular type of animal pollinator or abiotic agent like wind or water. Now, most Penstemon species are adapted to be pollinated by bees, and there's been separate evolutionary transitions to a hummingbird pollination syndrome. And this involves change in multiple floral traits. So bee-adapted Penstemons tend to have blue or purple flowers, a color that stands out well to bees. They produce small amounts of concentrated nectar. The lower petal forms this landing platform for bees, formed by the lower petal lobe. And these flowers are relatively short and wide, allowing a bee or wasp to fully enter the flower, access the nectar reward at the base of the flower, and contact the anthers and stigma that are located within the flower. And then the hummingbird adapted penstemons have evolved this bright red flower color that stands out well to hummingbirds. They produce lots of nectar. And of course, hummingbirds have large energetic demands, and they're not going to be reliably visiting those flowers that don't produce that large nectar reward. They tend to lack the landing platform formed by the lower petal lobes. This is either sort of reflexed back or reduced, and they have this narrow tubular shape. And the stamen filaments and style are elongated so that the anthers and stigma are outside the flower, where they'll come into contact with the forehead of visiting hummingbirds. And so if we think about a transition from bee to hummingbird pollination, most likely the first traits to transition would probably be these attraction and reward traits. And then maybe you would get selection to improve the morphological fit between the pollinator and the flower and increase the efficiency of pollen transfer. Okay, so to understand the pattern of floral evolution in Penstemon, we need a phylogeny, and we previously took a phylogenomic approach and sampled a large clade within Penstemon that included about 1/3 of the total species diversity. And for this, we generated our genetic sequence data using multiplex shotgun genotyping, which is a type of rad-seq. And here I've marked the hummingbird syndrome species with red dots, and the other species all tend to show the ancestral bee pollination syndrome. And so what you'll notice is that these hummingbird syndrome species are distributed across the tree. Most seem to represent a separate origin of this floral type, and they're sister to bee-adapted species. So this suggests there's been many shifts from bee to hummingbird pollination. Just in this sample, there are 16 origins. And we don't see the repeated evolution of other floral syndromes that are common in North American flora. So we don't see, for example, repeated shifts to hawk moth pollination or wind pollination or self-pollination. And this is what's really compelling about Penstemon, is that it shows this widespread but highly specific pattern of repeated evolution. So maybe there are genetic features that basically predispose Penstemon to this specific shift, even though it requires, obviously, complex change in multiple traits. So my talk is going to be divided into 3 parts. So first, I'm going to briefly discuss some macroevolutionary modeling results where we modeled these transitions between these floral syndromes. Then I'm going to discuss the genetic architecture of floral divergence in a focal species pair. And then finally, I'm going to discuss some population genomic data that informs on how these floral differences are maintained in the face of gene flow. All right, so it seems that these shifts from bee to hummingbird pollination have been frequent within Penstemon, but hummingbird syndrome remains rare overall. We don't see clades of hummingbird adapted species. So maybe there just hasn't been enough time for these hummingbird syndrome lineages to have diversified and reach greater numbers. Or maybe hummingbird syndrome itself is somehow associated with a reduced speciation rate or reduced diversification rate, which could be driven by reduced speciation or increased extinction. So we explored these hypotheses using the VISI model of trait-dependent diversification. So this model jointly models the tree and binary character data. So in our case, bee versus hummingbird pollination, and it fits 6 macroevolutionary rate parameters. So these are speciation and extinction rates associated with each of the 2 character states, as well as the rates of transitioning between these. If you happen to be familiar with these models, you're probably aware there are more complicated versions that include additional hidden character states. And we did explore these models, but this simpler model was actually a better fit to our data. And so we estimated our parameters using a Bayesian framework, giving us posterior distributions of parameter values. And we have two results from this exercise. So the first concerns asymmetry in the transition rates. So we found that the rate of transitioning from bee to hummingbird pollination is shifted towards higher values than the rate of transitioning in the reverse direction, which is estimated near zero. And here I'm showing an ancestral character reconstruction where I've colored internal nodes according to the posterior probability of being bee or hummingbird syndrome. The second result is that the hummingbird syndrome is associated with a substantially reduced diversification rate that seems to be driven by a reduced speciation rate. Now, there are a lot of caveats to these state-dependent diversification models. So we know that the dynamics of diversification are much more complicated than are modeled by these relatively simple approaches. And, in many situations, there could be multiple sets of parameter values that are equal fits to the data. So one way we can kind of validate these models is to connect these broader macroevolutionary patterns to microevolutionary processes at the population level. And so we're really interested in using these models basically to generate a hypothesis that we'll then investigate with population genetic data and understand this pattern of repeated kind of asymmetric shifts from bee to hummingbird pollination and explore the consequences of that transition on potential diversification rates. So just really briefly, this potential difference in diversification rate is really compelling, and it kind of leads to some hypotheses on underlying mechanisms. And so one that I've been thinking about is that maybe pollination by hummingbirds actually increases genetic cohesion between populations, because maybe hummingbirds are moving pollen greater distances across the landscape. And in theory, that increased genetic connectivity could reduce the opportunity for allopatric speciation, basically. And so a prediction of this would be that perhaps you should see greater genetic variation segregating within populations of the hummingbird pollinated things, as well as greater shared variation between populations. So we'd like to investigate this in the future, but for now we will leave this diversification rate finding behind and focus on this high rate of transitioning from bee to hummingbird syndrome, and just kind of this overarching question: how are these floral differences established and maintained in these recently diverged species pairs that we see in Penstemon? And what's nice is that we do have these nice replicated sister species pairs, so we can use a comparative framework to address this question. Here I've zoomed in on a focal clade that includes 4 separate origins of hummingbird adaptation, which is a really nice place to start. And for the past few years, I've been heavily focused on this species pair here to begin to understand the genetic architecture of floral divergence. And these are Penstemon barbatus and Penstemon virgatus. They're sister species. They're pollinated by different pollinators, and they show that divergent floral adaptations. And they have overlapping distributions in the Southern Rockies and in the Sky Islands of Arizona and New Mexico. In some places, they can occur sympatrically. And we previously crossed these species to generate an F2 population for QTL mapping. So we measured floral traits associated with this pollination syndrome difference. And then we generated genetic data, again, using multiplex shotgun genotyping. So this gave us a set of markers that we ordered into a linkage map and used for QTL mapping. And we found that flower color and nectar volume each involve a single locus of major effect, both of which are located on our linkage group 1, and the peak LOD positions are about 10 centimorgans apart. Now, these two traits are really critical for initially attracting hummingbirds, and I think they could be important first steps during a transition from bee to hummingbird pollination. And so it's notable that each of these traits may have only required a single mutational step. Now, for one of these traits, flower color, of course, we can really dig in to the molecular genetic basis and identify the mutations that contribute to adaptation. And this is because, flower color in Penstemon, as in many plant species, is the result of anthocyanin pigmentation, and the anthocyanin pathway is highly conserved across plants. So we can use a candidate gene approach to address this question. So here I'm showing you a very simplified version of the anthocyanin pathway that just shows some of the enzymatic reactions that we're interested in for this talk. Okay, so we know that shifts from bluish to red flowers in Penstemon involve a shift from producing these blue delphinidin-based pigments to producing red pelargonidin-based pigments. And producing delphinidin requires the activity of this branch point enzyme called flavonoid 3' 5' hydroxylase, or F3' 5'H. And we found that flower color in our F2 population co-segregates with genotype at F3' 5'H. And it turns out that the red flowered species, Penstemon barbatus, has a non-functional copy of this gene that's been inactivated through at least 2 loss of function mutations to the coding sequence. So one of these is a premature stop codon that truncates a heme binding domain, and this stop codon is sufficient to abolish enzyme activity. But when we reverse that stop codon through site-directed mutagenesis, we don't restore activity. So there must be another additional inactivating mutation elsewhere in the coding sequence. And it turns out that this type of loss of function mutation to this gene, F3' 5'H, is a common route to red flowers in Penstemon. So we looked at this gene in 12 additional origins of red hummingbird adapted species and find that it's predictably inactivated in nearly all of these species. We see a variety of premature stop codons, frame shift mutations, and large deletions. So this is just showing kind of a schematic of the coding region. And in many cases, the gene is like extensively degraded. And of course, once an initial mutation inactivates the enzyme, it's going to accumulate additional mutations on its way to becoming a pseudogene. Okay, so why are coding mutations to this gene such a predictable outcome? Well, we know there's a large mutational target size for loss of function because there's many ways to break a gene. And also, mutations to this gene don't disrupt the production of other flavonoid compounds that are important for plant fitness, compared to other ways you could like mess with this pathway to get a shift to red flowers. And so, this large target size may help explain why shifts from blue to red flowers are apparently so genetically accessible to these Penstemon wildflowers. But these redundant inactivating mutations that accumulate could generate a genetic constraint on reversals from red flowers back to blue flowers, because you'd have to repair those multiple loss of function mutations, which could be quite unlikely. And it's possible that this kind of genetic constraint actually contributes to this asymmetric transition rates. Okay, so back to our QTL results. I told you about our major effect QTLs for flower color and nectar production. It turns out that there's another trait that involves a single QTL of major effect, which is flower width. And, you know, these three traits together really define hummingbird syndrome in Penstemon. And so, you know, I'd like to suggest that this is a relatively simple genetic architecture that might streamline shifts to hummingbird syndrome in Penstemon barbatus. Now, other aspects of floral morphology were not genetically simple. So these were more polygenic, involving smaller effect QTLs. And here I'm just showing the LOD trace for style length as an example. Okay, so we have recently generated a de novo genome assembly for Penstemon barbatus, the hummingbird pollinated species. So this means that we can identify the physical locations of these floral trait QTLs. And I wanted to just give you a quick piece of information about our genome assembly. So this is a chromosome level assembly that's supported by our linkage map from our F2 population. And we see that each chromosome has an extended region of low recombination. So these green traces are showing genetic distance in centimorgans as a function of physical distance in megabases. And so these plateaus are the regions of low recombination. And we find that these low recombination regions are basically depleted in genes compared to the high recombination regions. And so this orange histogram is showing the density of genes along each chromosome. And so we can overlay our QTL peak LOD positions onto this plot. And we see that our floral trait QTLs are mapping to the gene-rich high recombination regions, which, you know, makes sense. That's where the genes are for the most part. And they're fairly distributed across the genome, although we do see some areas where few traits are clumped together, but there's still recombination between them. Okay, so I've suggested that perhaps this relatively simple genetic architecture may have streamlined the evolution of hummingbird pollinated traits in Penstemon barbatus. So how are these floral differences maintained in the face of gene flow? And this is relevant because, you know, these two species do have overlapping distributions and they can occur sympatrically. So to examine patterns of genetic divergence between these guys, we sampled several populations of each across their range, sampling 8 individuals per population. And we basically generated shallow genomic data, again, using multiplex shotgun genotyping, but this time using a really frequently cutting restriction enzyme. So we align these data to our new reference genome and called genotypes, which gave us shallow coverage across roughly a quarter of the total genome length. And then we calculated pairwise genetic distance between all individuals in the study. And we can visualize average genetic distance between populations as a neighbor joining tree. And taken at face value, it describes this interesting result, which is populations of the hummingbird adapted species barbatus that occur out east in Colorado and New Mexico are actually more closely related to the nearby eastern Virgatus populations than they are to barbatus populations out west in Arizona. And we can also visualize this pairwise distance matrix in 2 dimensions using multidimensional scaling. So this allows us to kind of overcome that assumption of a bifurcating tree. And here, each dot is representing a population, and the absolute distance between points is reflecting the average genetic distance between those populations, okay? And so we recover the same pattern where these Eastern Barbados populations are closer to the Eastern Virgatus populations than they are to Western Barbados populations. So here is a distribution of pairwise DXY between populations of the hummingbird adapted species shown in this red plot, and the interspecific population pairs shown in the light green. And we see substantial overlap between these two categories. And the results are similar for comparing the distribution of pairwise FST. And this overlap, again, reflects the fact that nearby populations of different species can be more closely related than distant populations of the same species. And here I'm just showing those pairwise genetic divergence as a function of geographic distance, and the same for pairwise FST. Mainly to point out that it's not only the within population comparisons, but also the between population comparisons show a positive slope. Okay, so all of this suggests that there's been substantial genetic intermingling between these two species at sort of a regional scale, despite them being strongly differentiated in their floral traits and in their pollinator. And so, you know, we'd like to know where in the genome these two species are strongly differentiated. In other words, what parts of the genome are holding Penstemon barbatus together as a recognizable and unmistakable species? So we started by looking for fixed differences, like range-wide fixed differences between these guys. And we really came up short. So there are very few SNPs that are fixed differences between species. Most of the biallelic SNPs in our data set are polymorphic in both species or segregating in either barbatus or virgatus. And the sites that do appear fixed actually have low coverage across individuals. And so they may appear fixed just spuriously due to a small sample size. So given this lack of fixed differences, we conducted a SNP by SNP test for an association between genotype and species identity using a likelihood-based approach that can handle genotype uncertainty. So we calculated the likelihood that each of the three possible genotypes at a given SNP show a distinct probability of being found in Penstemon barbatus versus virgatus. And so that would indicate an association of SNP genotype with species identity, and compared that to a null hypothesis of no association using the likelihood ratio test. And so here I'm showing you these SNP associations. So higher values of the likelihood ratio test statistic, or LRT, indicate a strong association between genotype and species identity. And so we do find strongly associated SNPs throughout the genome in both the high recombination regions and these low recombination regions that I've indicated with the gray shading. And we know that these SNP associations are strongly predicting difference in allele frequency between the two species. So the SNPs with the highest likelihood ratio test statistic are nearly fixed between the species. And we can consider a set of nearly fixed SNPs with high significance as a set of species diagnostic loci, okay? And these outlier SNPs tag about 20 regions throughout the genome. They're sprinkled throughout the genome, and of course, we know the positions of our floral trait QTLs, so we can add those on the plot. And we see actually some of our highest SNP associations with species identity are located right on top of those floral trait QTL peaks. Okay, so I want to zoom in on a couple examples. So here on the left are those linked flower color and nectar volume QTLs on our chromosome 1. And here on the right is our flower width QTL on linkage group chromosome 4. So again, these are some of the highest SNP associations in our data set, and they're right on top of these peak LOD positions for floral traits. What's surprising is we don't actually see local peaks of DXY or FST surrounding these outlier regions. So levels of DXY and levels of FST between the species aren't strongly elevated above the genome-wide background. So these major effect floral syndrome loci are not really detectable as a deviation from the common pattern of genetic differentiation. Instead, it seems like divergence between these floral syndromes involves these fine-grained SNP differences in these high recombination regions that just don't leave an extended genomic signature. So just to give you kind of a sense of what this data looks like, here I'm showing some genotype tables for some of the strongly associated SNPs. And the blue values are the genotype tallies for the bee adapted species, and the red values are for the hummingbird adapted species. And so, these strongly associated SNPs are effectively in linkage disequilibrium with each other, but not with these neighboring SNPs that are unpredictive of species identity. And so it seems like a pretty small part of the genome is actually diagnosing the species in this case, or what we're calling the species. Okay. Now, what's nice is that these SNP associations actually help us narrow down our QTL intervals. In fact, we've seen some really interesting candidate genes near these floral QTLs. So strongly associated SNPs near our nectar volume QTL are right next to this basic helix loop helix transcription factor. And members of this are known to affect the patterning of trichomes. And Penstemon nectaries are a patch of glandular trichomes. And so we're interested in this as a possible candidate gene. And then for the flower width locus, we see strongly associated SNPs are right next to an apetalate 2 homolog. And this transcription factor has known effects on flower shape, so it's a really good candidate for us as well. Okay, so we think this species complex is kind of ideal for studying the origin and maintenance of these complex floral differences, because despite their strongly differentiated floral traits that kind of immediately are recognizable and we can immediately diagnose the species by looking at it, the majority of genome-wide variation does not predict species. And so it's well suited for association mapping, even using structured population sampling. So in the future, we're going to kind of resample and use whole genome resequencing so we can gain a much higher resolution view of what's going on and follow up with some of these candidate genes, and then start to examine whether we can see any signatures of selective sweeps, and if so, whether there's evidence that the strength of selection differs for these genetically simple traits versus the more polygenic traits. And then kind of stepping back, this work kind of fits into our larger efforts to study the comparative genetics of floral divergence in Penstemon and begin to understand whether this relatively simple genetic architecture that we see in this species pair is a shared feature in Penstemon, maybe explaining why these shifts have occurred so many times. We're also interested in the source of genetic variation for these repeated shifts. For example, is there any evidence that introgression has moved hummingbird adapted QTLs between lineages? And then we're also interested in like the consequences of hybridization between these two floral syndromes. So for example, what are the fitness effects of intermediacy between these two syndromes? Now, Penstemon barbatus and virgatus actually don't show extensive hybridization in the field. I have seen hybrids, but they're not common, which is really interesting, given the amount of shared variation that we see. But in the last little bit of time, I'll tell you about a second species pair that we're diving into. So this is another one of the pairs within this focal clade. This is Penstemon eatoni, that's hummingbird adapted, and laevis, that's bee adapted. Eatoni has this broad distribution in the Southwest, and laevis is this narrow endemic. And so we've been sampling populations in this area of sympatry. And we have just like a pilot data set where we sampled individuals from a couple populations of each species in this area and generated whole genome resequencing. And to start out, we've been calculating DXY between these populations to look for any peaks of differentiation. And again, we don't see obvious peaks. And I know this is a little bit of a nightmare to look at, but basically I'm showing you pairwise DXY between populations of the hummingbird adapted species, that's the red trace, and between the two species, the dark blue trace. And these lines are nearly right on top of each other. And also, by the way, there are very few fixed differences between these guys. So clearly there's a lot of gene flow in this species pair as well. In fact, actually, they show these like spectacular hybrid zones where they come into contact. And in the past couple of years, we've been spending a lot of time in these hybrid zones in these canyon systems on the Arizona Strip, where we see just a huge amount of segregating variation in floral traits. So in work that's funded by the NSF, we are going to use admixture mapping to localize the genetic basis of the segregating variation, and then also hopefully study patterns of selection, natural selection acting on these hybrid individuals. And so this is work that involves Josh, a grad student in my lab, and Hoyt, a research technician. All right, so this brings me to the end. I'm eager to get you guys' feedback. Lots of thanks to our collaborators, particularly Lena and John at University of Kansas, where this work kind of got launched, and then resources at Kansas, South Carolina, and our funding. So with that, I'd be happy to take any questions. Okay. Yes. Oh, is your question which pollinator is? We have, okay, so the question is, who is pollinating the hybrids in our hybrid swarms? We have not done extensive pollinator observations. I have seen hummingbirds and bees pollinating these guys, but we haven't looked for associations between traits and pollinators. And to be honest, that would be like really time consuming. So I'm not sure we'll do that in the near future. It's a good question. Yes.
You had kind of described most of them as being an endpoint of their being pollinated or
yeah. Great question. So the question is, are there any populations that are sort of intermediate between bee and hummingbird pollination? So in the focal clade that I was describing that has those 4 origins of hummingbird pollination, really it's effectively a binary trait. However, there are some species of Penstemon that do show what I would call intermediate floral syndromes and maybe are more of generalists. Interestingly, these seem to be, there seems to be a geographic correlation with those. They tend to be in Southern California. And they're also, I would say, phylogenetic related to each other. But these guys have like basically hot pink flowers that aren't quite as narrow. And I'm not sure about the amounts of nectar. Nectar is kind of a tricky thing to like measure in the field. And there's been a bit of pollinator work on those that show that they are visited by both. And it's interesting to think, like, is that an endpoint? Is that kind of a stable situation? Or are they on their way to full hummingbird adaptation? It's a good question. Okay, I'll start with you.
I was curious about other flowering phenomena. Are they like largely overlapping or are they separate at all? Are they at the same time?
That's a great question. So for the Barbatus virgatus that I spent the most time talking about, they do flower at the same time. However, Barbatus flowers over a much longer period, the hummingbird adapted species. And with the second species pair I briefly mentioned at the end, they also have overlapping. Now, it's possible that they're slightly shifted. And I don't have the data to tell you for sure, but that second species pair, they flower early in May, and basically they have more similar flowering periods. Like the hummingbird pollinated species is flowering for roughly the same duration as the bee pollinated species. Yes.
Do you have a sense for what the range of these guys look like over the last glacial period, like last 100,000 years? Like I'm wondering, you know, elevation now, if they were lower elevation, maybe quite spread.
Okay, so the question was, do we know the historic geographic range of these guys? No. And I know that there are methods to reconstruct these things based on current occurrence data and then kind of project things back. And we haven't done that for these guys, but it would be interesting to know. Yeah.
Just see like a decline in effective population size or anything like that related to like climatic transition. I'm just wondering if it's high elevation populated underwent the extreme bottlenecks during that.
Okay, so the question was whether we see any differences in effective population size sort of shaped by range expansions, for example, increasing in elevation or something like that. That's really interesting. And I don't know. I think that could be, you know, I don't have enough data to associate genetic variation with like elevational differences. I've thought a bit about how elevation could shape patterns of genetic differentiation, because things that are at higher elevations are flowering later. And so that could be restricting gene flow between populations at different elevations. But yeah, I haven't thought about that other part. Yes.
Really great.
Yeah.
I was wondering with your phylogeny, you were showing with the leaves that have the hummingbird, have you done any work to figure out like exactly when that transition happens?
So the question is, have we determined the timing of the transitions to hummingbird pollination. No, and I think it's tricky unless we knew definitively the genes involved in that transition and could kind of trace the history of those loci backwards. I mean, we can impose some model of character transitions onto the tree and explore it that way, but no, we haven't looked at that. Yes.
Yeah, I think we're on something there. If you look at patterns of images, well, too many from your QTL study. It would look like you're, as you said, quite narrow regions of association. That's indicative of really cool rather than young.
Yeah.
So it could very well be that a lot of shared here in the and separately out various things. So dating some of the potential could be really, really.
Yeah, exactly. Because we have, sorry, the question, well, it was more of a comment than a question, which was, we do have actually this valuable information from our population genetic data. We have these like very narrow peaks of divergence that suggest there could be ancient segregating variation that's experienced extensive recombination and erosion of the original haplotypes on which those hummingbird adapted traits arose on. And yeah, that's actually a great point because we do have the QTL data for this cross. And, perhaps we'll actually be successful in our admixture mapping approaches for the second species pair. We also have a genetic cross for that in the works, but it takes a long time to do genetics and perennial wildflowers. So as a slow burn, but yeah, so we could actually, target the age of those alleles. And I think that's actually a really interesting, follow up.
So, and ask that question. So here's the question. So some of these proposals that you have, you show us the Penstemon tree, and at least one plate on the far left, where it's clearly exceptionally red, and when you were forced to what have you sequenced F3 prime, 5 prime in reversal situations?
So let's look at that. So the question was, okay. So you're thinking about,
Yes, right there.
well, yeah, we should. So the question was, we have a small clade that appears to be, so basically there could be an evolutionary reversal to bee pollination, or there could be 3 separate shifts to hummingbird pollination. And the first scenario is more parsimonious or more intuitive if we look at this. I haven't followed up on that, but that would be a great place to look. And, I'm a little bit agnostic on whether that would be a reversal or 3 separate shifts. That's so that little group of species are in separate little sky islands in Southeast New Mexico and far Southwest Texas. So there's like an endemic to Big Bend, there's an endemic to the Davis Mountains and things like this. So yeah, it would be interesting to look and see what's going on there for sure. Yeah. Sure. Yeah, so it's actually, right. So under the best model by the BISI model, which of course has these asymmetric shifts, and we do see basically infers the ancestor of this pair is most likely be adapted, and then maybe the shift to hummingbird pollination occurred maybe on this branch. But it would be good to go into the genetic data and see what's going on at F3'5'Ah. Matt.
You alluded to it, but I just wanted to sort of get you to follow up on it. Why do you think that these transitions have consistently happened to hummingbird pollinators and not for other pollinators?
Yeah, so the question is, why does Penstemon go to hummingbird pollination and not like hawkmoth pollination? So Penstemon flowers don't really make a floral scent, like a strong floral scent. And I think that could be a constraint on shifts to hawkmoth pollination. Why haven't they gone to self-pollination? They're perennials. And there aren't many selfing perennial species, right? If there's a period of, even if a plant is obligately outcrossing, okay, and there's a year where their pollinators just really aren't around, they might be able to make it to the next year and then have some fitness. And interestingly, nearly all hummingbird adapted species in North America are perennial, like those go together, which is something that I'm kind of interested in, the link to perenniality. And that's a longer conversation on why I think those could go together. But yeah, did that answer your question? Okay. Guys.
Is it BYOT or it will be up there?
Oh, there won't be T, but I do like cell service.